Uh, recording. All right. Hi, my name is Josh. I'm a PhD student in oceanography, and yeah, today I'm going to be talking to you about my research rotation at NASA Goddard, where I focused on agnostic biosignatures and taking a recently proposed theory for measuring these called molecular assembly, and then and trying it out with a new approach that's really well validated for spaceflight called gas chromatography mass spectrometry to see if we can measure what the theory predicts in order to use that as a biosignature. So to start with this title, biosignatures are the way that astrobiologists search for life. So one of our big goals in astrobiology is that we're looking for potentially life on other worlds. And to do that, we are looking for biosignatures, which can be a lot of different things. And until recently, a lot of those have focused a lot on what Earth life looks like. So Earth life is composed of proteins and amino acids and DNA and lipids. And we've been looking for a lot of things that are similar to those fundamental components of Earth life when searching for biosignatures on other worlds. However, recently there's been a big push towards agnostic biosignatures, where agnostic biosignatures are ones where we stop making those assumptions about Earth life and its fundamental aspects and think about, in a bigger picture, what life means and what it means to be alive. And so I think this is really well phrased as the search for life as we don't know it, instead of searching for life as we do know it. So with these biosignatures that are agnostic, we're trying to stop making these assumptions about the biochemistry that this life would be using. And some examples of this include biosignatures taken from a range of different fields, including atmospheric chemical disequilibrium that we can measure in an exoplanet, non-random motility that you can measure if you're watching a bacteria swim towards a higher concentration of nutrients, or in my case, as an organic geochemist, thinking about the chemical complexity of the organic molecules that we're measuring. And a lot of the work I'm going to be talking about today is inspired by this paper by Marshall et al. In 2021 called Identifying Molecules as Biosignatures with Assembly Theory and Mass Spectrometry. And so within this, I want to break down what assembly theory is and what mass spectrometry is, because I know a lot of you do not spend as much time with mass spectrometers as I do, and how we can use these two components and pull them together to look for biosignatures. So assembly theory is an approach for quantifying the complexity of molecules that relies on graph theory. And so within molecular assembly theory, you can build up molecules from individual atoms by adding in bonds or subfragments or additional atoms as individual steps. And you do this in the simplest way possible, and that gives you a number of steps. And you can count that number of steps, and that gives you essentially a number you can associate with the complexity of the molecule. So if we look at something really small and simple, like asparagine on the bottom here, it was built in 7 steps. It doesn't have too many atoms or too many different types of bonds or different types of substructures. So it's a relatively simple molecule. However, when we get up to something like ATP there in the top, that is a molecular assembly of 21, it takes a lot more steps to build that molecule. And this is really useful because it allows us to take complexity from this kind of vague, amorphous, qualitative characteristic and turn it into a quantifiable characteristic. The next part of that title involved mass spectrometry. And I'm going to be talking about the type of mass spectrometry I do as an organic geochemist. And that is where I'm measuring organic molecules. And so to do this, I start with a sample that's going to be a mixture of complex organic matter that's a mixture, which means it has lots and lots of different organic molecules in it. We then do a separation step called our chromatography, where we take that mixture and we spread it out as much as possible. We separate it into as much as possible, and that allows us to measure each thing individually. And by measuring them individually, we can measure them with a much higher degree of accuracy to a lower limit of detection. We're just a lot better at measuring things when we're measuring things one at a time. We then ionize our sample because we need it to have a charge in order to make a measurement on a mass spectrometer. And then we detect it, where we're measuring the mass to charge ratio, so the mass kind of on our x-axis, and then we get a signal intensity, which is directly related to how much of that compound there is. We can also sometimes do an additional step known as fragmentation or tandem mass spectrometry, where we take those molecules that we're measuring and then we break them apart by hitting them in what's known as a HCD cell that hits them with a neutral ion. It causes them to break, and based on how they break, we can learn more about the molecule because we're measuring the different types of fragments it's made up of. So in organic geochemistry, there's kind of two main types of mass spectrometry that use different separation and ionization techniques in order to measure the molecules. The first one is gas chromatography, or GC, electron ionization, EI, mass spectrometry. And so this separation is in the gas phase, so we need our compounds to be volatile, and it uses a hard ionization technique called electron ionization, where you blast the molecule with electrons, and that causes it to fragment. That's the hard part of it. We are breaking it apart, but then all those fragments pick up charges, and we measure all of those different fragments. The other main type, and the type that has primarily been used with molecular assembly, is liquid chromatography, electrospray ionization mass spectrometry, which is a long name, but essentially we can break it up into its parts where the separation is in the liquid phase instead of the gas phase, so we don't need our analytes to be volatile. The ionization technique is now softer, so instead of breaking that molecule apart, we're kind of having the charge go directly onto the whole molecule and it doesn't fall apart, allowing us to measure the entire molecule at one time. And then, yeah, so those are kind of our two types. And you can see two examples of the different types of columns that are used in this separation. So on the left, we have the long, spindly GC columns inside of an oven because you use temperature primarily to drive the separation. And then on the right side here, we see these kind of short, stubby, bulky liquid chromatography columns that are built to withstand really high pressures that we put them under in order to force compounds and liquids through. So now to walk you through what fragmentation looks like in ionization for these two different types of mass spectrometry approaches. So we start with a molecule. In this case, it's just a random example where we have a green circle that's one atom that weighs 5, a purple circle that weighs 10, and an orange circle that weighs 20. The whole mass of that molecule is 35. When we use gas chromatography, in this case, we're using our hard ionization technique of electron ionization. It breaks into lots of different fragments. And this is the only way we can measure it. We can't measure that whole molecule at one time. And that gives us a mass spectra like I was talking about here, where we have our mass to charge ratio on the x-axis and then signal intensity on the y-axis. And each of these fragments corresponds to one of the peaks we see in the mass spectrometer. So that green 5 is that 5 on the mass spectra plot. We can then continue on and do that fourth extra step, that tandem fragmentation like I was talking about, with the HCD collision, where we select a fragment, in this case, our mass 15 green and purple fragment, and break it apart. And then we measure the mass of those fragments. So this is essentially the data we get from a GCMS. LCMS, on the other hand, has some differences. So we're once again starting with our same initial analyte. And if we use the electrospray ionization technique, it's that soft ionization technique. So instead of 5 different fragments, none of which are the whole original molecular ion, we get just one peak, which is specifically related, which is just the mass of that initial ion. And then we can do that same fragmentation and get a similar fragmentation pattern to what we got with the GCMS MS1 data. So the main takeaway I want you to get from this is that GCMS and LCMS are really not interchangeable, and that they have really important differences in terms of the type of data they produce and what you can analyze. There's often no molecular ions, so no single ion with GCMS data, and you often are you can measure different types of molecules, there's different types of collisions that will produce different types of fragmentations, and that GCMS often also requires derivatization. And derivatization is where you do chemical reactions to your analytes. You have to add pieces onto them to make them volatile enough to fly on a GCMS column. And so that can be a big drawback because you're fundamentally changing what you're measuring. So now that we have a good solid foundation in mass spectrometry, we have a solid foundation in molecular assembly, how do we then bring those two together to estimate, use mass spectrometry to estimate molecular assembly? The way they did this in the Marshall et al. Paper is through using those fragmentation patterns. And it's actually a really simple idea where essentially the more complicated of a molecule you are, the more different fragments you are going to make. So when we start with a really simple molecule like succinic acid here, with a molecular assembly of 5, and we fragment it, we get 3 fragments. This is not a lot of fragments. However, as we start to go up and we get to molecular assemblies of 15 and 27, we see that our fragmentation spectra has a lot more fragments in it. And we can just simply count that number of fragments, and that gives us an estimate of complexity, where we look at the correlation between the total number of mass MS2 peaks that we're seeing, so the total number of unique fragments we're measuring in a molecule, and then compare that to the molecular assembly, and we get this really strong positive correlation across over 100 different molecules. Then taking that relationship that we've now determined, we can use that to predict the molecular assembly of an unknown compound. In this case, if we take our MS1 peak here, so we pick one molecule and we fragment it, and then we count the number of fragments, we can then use that relationship we figured out in the last slide to estimate the total molecular assembly, allowing us to kind of estimate the complexity of an unknown molecule. So now we have molecular assembly, we have mass spectrometry, we put them together. Now we have that final piece of doing this as a biosignature for life detection. And so I want us to think about how complexity is going to be distributed within a mixture of organic molecules. So if we take a distribution where complexity on the x-axis and abundance on the y-axis, we're going to get some distribution where there's going to be a lot of not very complicated things and very few complicated things when we have a non-living sample. And then for our instrument, we're always going to have some limit of detection. And this is going to be the amount of compound we have to have present in order to make a measurement. It's going to be somewhere on this abundance plot. However, when we have a living sample as opposed to a non-living sample, the distribution is going to change. We're going to have a lot more complicated molecules than we do in a non-living sample. And so in theory, what we can do is identify some sort of complexity threshold where we're only going to see molecules of a certain level of complexity above our limit of detection when we have living samples as opposed to non-living samples. And within that space, if we measure those molecules above that limit of detection and above that complexity threshold, that should be a piece of evidence pointing towards life. However, molecular assembly, while it sounds great, is this cool agnostic biosignature that's not assuming anything about the biochemistry present. It is nowhere near ready to go to space. It is not going to be going to Enceladus or Europa anytime soon because there's a lot more work to be done. There's currently one paper out about it. And so my work was to help move it along. And the main way I did this is by trying it with gas chromatography mass spectrometry. So molecular assembly has only been performed with LC-MS, which we know from what I just talked about is really different from GC-MS data. We're not getting those same molecular ions, the fragmentation is different. There's a lot of key differences that we have to consider. However, GC-MS is what's known as a heritage instrument. That means it has gone to space a lot. It has been tested in a lot of different facilities and a lot of different conditions, and we've gotten really good at doing GC-MS in space. However, molecular assembly has never been tested on it. And then in addition to this key difference of switching instrumentation, there's also a lot of other analytical factors that we think about here on Earth that haven't yet been considered with molecular assembly. These include, how are we preparing this sample? So there's a lot of different ways to get organic molecules out of just a sample of dirt or seawater or something like that. And that's a really important thing to consider because you're going to get really different compounds and mixtures if you do different sample preparation approaches. The chromatography needs to be optimized, the quality control parameters need to be considered, and we also have to think about compound concentration, and are we working at that right limit of detection like we were seeing in those other plots? So my two main questions I went into this with were, can GCMS be used for estimating molecular assembly? And what analytical factors are important to consider for further development of molecular assembly for future mission? So I wanted to identify key areas where we see high levels of variability that we need to be thinking about. And to do this, I traveled to NASA Goddard in Greenbelt, Maryland. So I spent my winter in DC and working at NASA, which was very cool. I got to see where they build a lot of the space telescopes. So you can actually see in this picture, the Nancy Roman Space Telescope being built in the world's largest clean room. I got to see the chambers where they essentially simulate space in order to see whether these instruments can handle being put in vacuum and cold temperatures and high pressure, things like that. It was just a really cool experience. And I worked with the Dr. Heather Graham and her Agnostic Biosignatures Comrades Lab, which they didn't tell me that's what it was called until like week 10, but it was cool. And yeah, so I worked with Dr. Heather Graham, who's going to be giving a talk here later this quarter. The postdoc, Dr. Gabriella Weiss, was one of my main mentors, and I spent most of my time pretty much every day with Dora the Explorus, which is an Explorus Orbitrap mass spectrometer, and all good instruments have names. So this one was named Dora. So the first step in doing this is we needed to develop a pipeline to count fragments from GCMS data. We're not just getting a single MS2 fragmentation spectra, so we needed to find a way to pull out a fragment number or a number of fragments from a raw GCMS chromatogram. And so what that data looks like is we start off with a total ion chromatogram. In this case, we're looking at retention time, which is essentially our separation step, where different compounds are entering the instrument at different times based on the separation steps we're doing. Where each peak you see, where it shoots up to be a really high intensity on the y-axis, is a compound. However, when we zoom in, we see that each of these peaks is actually made up of a bunch of different masses. So these are all of those different fragments I was telling you about. So that one compound is breaking up into all these different fragments, where each one of those lines is a different mass corresponding to a unique fragment. So, but we also have a ton of noise and other instrument signal in this mix. So how do we pull out only the fragments that are related to our compound? To do this, we started by identifying our peak that was the most abundant fragment. And you can do this either manually or with various detection algorithms. We then implemented a noise floor threshold where we removed all peaks below or all data points below a certain threshold in order to remove instrument noise. We calculated a correlation coefficient between these various fragments and our most abundant fragment, or like our unique identity fragment for this compound. And then we removed isotopes. So in these molecules, we're measuring the exact mass. So if you have a 13C isotope as opposed to a 12C isotope, you'll see that on your mass spectrometer, and that's not actually a unique fragment. So we don't want to count that in our data. And then we counted them all up. So I then ran this using this pipeline. I analyzed a number of different types of compounds. I analyzed amino acids, which we're all familiar with. These are the building blocks of our proteins. However, to make them fly on a GCMS, you have to do a derivatization step because otherwise they are not volatile enough to measure. So in this case, we used a trifluoroacetic acid methyl ester derivatization, which essentially adds chunks on to the amino acid, which increases the complexity of the molecule, which was kind of an interesting change to make. I also analyzed a mixture of alkanes. So here we see octane, and these are essentially your simplest organic molecules, where they're just carbon and hydrogens single bonded together. So they tend to have pretty high molecular weights, but really low complexities because they're such simple molecules. And then finally, I had a mixture of polyaromatic hydrocarbons. You may be familiar with some of these because these are a lot of organic pollutants. And they are volatile enough to fly on the GCMS without derivatization, but they're a bit more complicated than our alkanes. So they kind of provided a nice intermediate. And when we look at this relationship between the number of fragments we get from our data pipeline and the molecular assembly, we see that there is a positive relationship. It's not super strong, but it is statistically significant, with an R squared of about 0.32. And it suggests that GCMS can be used to estimate molecular assembly. However, I will be the first one to say that there's a lot more work to be done here. I ran it with as many compounds as I could in my 8-week period, but we need compounds of much higher molecular assemblies in order to get to really validate this as an effective strategy. But overall, I think this is really promising for doing MA work with GCMS. So then I wanted to consider how some of the quality control parameters we were thinking about are going to impact the results of our data, or our data pipeline. So I looked at the impact of noise floor, which is that threshold that I set for how abundant a compound or how much signal we have to see for a fragment in order to count it. And when I did that, I found that the responses were really variable across our different compound classes. Where we see for noise floor, I ranged it from 5,000 to 100,000 in terms of signal intensity, and then looked at the number of fragments. And each one of these lines is that of a different color is a different compound across our 3 different compound classes: amino acids, alkanes, and PAHs. And what we see is that it's not really predictable. Like, some go down pretty substantially, others remain completely flat. And yeah, I don't have an answer for why this is happening exactly and why it varies across these different groups, but it does mean that we should be considering it as we move forward. And that selection point, that selection of a noise floor is going to be really important for calculating molecular assembly. I then looked at the MS2 data that we collected using this approach for these different compounds. So once again, the GCMF MS2 data is a fragment of a fragment. So this is different than the MS2 data we were collecting for the LCMS compounds that are in that Marshall 21 paper. And so to do this, I fragmented the 5 largest MS1 fragments for each compound. I then did a quality control threshold of a quality control noise floor threshold to remove noisy peaks that were in there. That's just instrument noise. I deduplicated the MS2 fragment list to only keep unique fragments. So that way we're not double counting a lot of things, which would artificially potentially increase the complexity, that score that we're giving it. And then I also, in order to once again be considering what instrument parameters are important, I fragmented these across a range of HCD cell collision energies, ranging from 10 to 100 electron volts. So for starters, when we add up all of these MS2 fragments together for our largest 5 largest fragments, we don't see much of a relationship between combined number of fragments on our x-axis and molecular assembly on our y-axis. So we don't see anything terribly useful there. However, there was interesting relationships between the total number of MS2 fragments we collected and the HCD cell collision energies that varied a lot with the type of compound we were looking at. So for amino acids and alkanes, when we look at combined number of fragments on the x-axis and then our different HCD collision cell on the x-axis, combined number of fragments on the y-axis, we see these really sharp drop-offs as we increase the collision energy. Which means that the lower collision energies are giving you much higher numbers of fragments. However, for the polyaromatic hydrocarbons, the PAHs, we see this peak kind of at about 45 electron volts, where both the lower energies and the higher collision energies are giving you lower numbers of fragments, which means that the number of fragments we're getting from any of these collision energies is really variable based on the type of compound you're analyzing. And that means that you need to be considering that with your instrument design. But it's also potentially predictable as it seems to vary consistently within each compound class. I also want to say that while the MS2 data on its own might not have been useful for predicting molecular assembly, there is more work being done to combine MS1 and MS2 data for LCMS. And this could potentially, the work I've done here could potentially be built into future recursive algorithms that are being developed to estimate the complexity of molecules with better approaches than just doing a correlation between molecular assembly and total number of fragments. So finally, I applied this GCMS molecular assembly pipeline to some environmental samples. So I looked at one sample that was alive, which was spirulina extract, where spirulina is a cyanobacteria, so pretty much as alive as you can get. And this, I analyzed a sample that was derivatized using the amino acid derivatization method. And then I also analyzed some Murchison meteorite extracts. And this was definitely on my bucket list to do as a scientist at some point, was to analyze an extraterrestrial meteorite sample. So this was very cool. And I analyzed two different types of extracts from it. One was a DCM extract, so that's going to get us our more nonpolar things, and then an amino acid derivatization approach, like the one we were doing for the spirulina. And when we put it through the pipeline, what we see is that our living sample in blue, the spirulina amino acids, was higher than both of our Murchison samples, but not by much. And so I don't want to put too much weight on that. However, what I do want to emphasize is the difference between the two Murchison samples, where we're seeing really different values, or not really different, but like definitely different values. And I think this is important to consider because it really emphasizes how important sample preparation is. Like what you're putting into your instrument from your space probe, inside your space probe, is going to make a huge difference in what you're actually going to measure. And so therefore, that step is critical for considering for agnostic biosignature method development. So in conclusion, we were able to develop a pipeline to use GCMS data and count fragment numbers for MS1 and MS2 data. And this could be potentially built into more advanced algorithms for estimating molecular assembly that are currently being developed. We found a strong correlation between GCMS MS1 fragments and molecular assembly, suggesting that we are seeing similar relationships to what we saw with LCMS data. But we need a lot more compounds of much higher complexity to verify these results. Finally, we highlighted the importance of analytical factors in the actual results that we're getting. So we need to be thinking about all of these different parameters as we continue on with our spaceflight mission development. And finally, we found that some of these analytical considerations were specific to certain compound classes, but some were not necessarily predictable. I also want to emphasize the different skills I gained. And I learned a new instrument on this research rotation. I learned a new discipline with planetary science and agnostic biosignature research. And I got exposed to a whole new set of considerations when considering how to put something in space and how much harder it is to do that. And I thought things were hard to do at sea, where you're on a moving ship, as opposed to in a lab, but going to space is just a whole different thing. I got to analyze a meteorite sample, which that's just cool. And I gained insight into careers at NASA and federal government labs. That was really valuable. So finally, I would like to thank Heather and Gabriella and the rest of the ABC's lab, as well as NASA Goddard and the Astrobiology Analytical Laboratory in which I sat there. And then I want to give a shout out to the UW Astrobiology program for funding this, as well as Lisa in particular for helping really make disappearing from my life here and going to a completely different place not horribly painful. She did a lot of work to help make that better. And I really appreciate that. And that's what I have. So thank you very much for your time.
Okay, so questions for Josh?
Yeah.
You have the slide showing like the different, like there's technical.
Or
Yeah, yeah.
this one?
So how exactly do you calculate the molecular assembly? Is it like each like organic synthesis step counts as one? Or
Yeah, so kind of every time you're adding a unique thing, it counts as one, but then you can add like entire chunks together. So if you're making like an alkane and you want to have octane, you could just add two butanes together. To CH4 plus CH4 gives you CH8, but that's only one step. That's not necessarily.
Why do you not count like the steps to get to?
Well, you've already counted those, so you've already added in a bunch of things, but you don't repeat steps.
Oh, it's a unique step.
Yeah. Yeah.
Is there a threshold of steps that's like potentially indicative of life, or is it just as complex as you can?
That I think is really up for debate. In this paper, they say 15, but their sample preparation steps are pretty highly variable, as well as a lot of other factors. So I think we don't know yet. Like, I think that is a really important thing to be thinking about, and it may be really dependent on the context, where you have some icy moons are full of complex organics. So it may be a much higher threshold there than in places where there's a lot fewer organics and a lot simpler organic chemistry happening. You may need a much lower threshold. So I think it may be dependent on the context. Yeah.
Kind of building on an earlier question, one big question I still have about assembly theory is like each of these individual steps, are these like at Earth temperatures and pressures? Like presumably on Mars or at the bottom of an ocean, on an icy ocean world, like the reaction pathways would be differently depending on like pressure, temperature, redox, whatever. So
Yeah.
if we're using this as like an agnostic biosignature, isn't it still like tied to kind of like Earth, the way that we build molecules here on Earth? And would, like, do we consider at all the different reaction pathways in different environments?
So I think the actual assemblies are based purely on the structure. So they're not actually considering what organic synthesis step would be required and like whether you need a catalyst or anything like that. I don't think any of that is built into it. I'm not an expert on assembly theory. I was mostly focused on the mass spectrometry and method development side of things. I don't think they're incorporating any of that information, but I can see that being really useful for considering what your complexity threshold would be. So if you're on, like you said, like high pressures, high temperatures, super cold temperatures, that's all going to make a difference with what types of molecules you're going to see and what that abiotic distribution you'd predict would be. So I think that's actually a really good point. But no, I don't think that's built in at all. Yeah.
I was just wondering about the repeatability of like counting the number of fragments, so if you run the same compound twice.
Yeah, so I didn't show that data, but it actually was shockingly reproducible. So with GCMS, a lot of what you get is these like fragmentation spectra, and people have built up huge libraries because those fragmentation spectra tend to be really predictable. And so you can use like, it'll give you sometimes 100 teeny tiny little fragments, and you can use that and their relative abundances to predict exactly what compound you're looking at. So I think it's pretty robust and reproducible. And when I ran my different algorithm across multiple files, I got really similar results. Yeah.
Eric has a question.
Oh, can you go to your spirulina versus Murchison thing?
Yeah.
Your graph comparison.
Yeah, and once again, don't put too much weight on that. It is preliminary.
Okay. So we were just having this discussion, right, about what MA do you need to be alive?
Mm-hmm.
So spirulina doesn't make it, it's not 15, so it's dead.
Yeah, but that was purely done with the LCMS data. So you may see very different things with LCMS versus GCMS in terms of what that threshold is.
Okay.
And that's also going to be dependent on your concentrations and your sample preparation. So I think there's a lot of work to be done on that threshold where they just kind of drew a line.
And my other sort of question would be, that's just one carbonaceous chondrite. There's many others. I mean, of course, Murchison's famous for having a lot of organics of 14,000 or something. But, maybe there are other ones that have a higher MA. And also, if you went to Titan, nobody thinks there's life on Titan because it's 90 Kelvin.
Yeah.
But there's a lot of complex organics, which they might score quite high on this, maybe even higher than spirulina. So I don't know, it seems very fuzzy.
I agree.
Okay.
And I think that's what I was hoping to show with my research is that,
Right.
or some of this is that, one, develop some of the initial pipeline, but then emphasize, there's a lot of work to be done and highlight where we should be focusing some of those efforts.
My other comment would be, it's called agnostic, but it isn't really, because the molecular assembly people have chosen one characteristic of life, which they've decided is really important, which is molecular assembly number. That's like, to me, the opposite of agnostic. It's actually zooming in on one thing and saying, this is what we look for. I know it's independent of the molecules, but it's not really agnostic.
Yeah, but I would say that, I mean, all of the ladders for life detection rely on lots of different signals pointing to the same thing. Like any individual biosignature, we would say, is probably not going to do it. It's not going to be enough to just measure one thing. You're going to need a lot of different pieces of evidence. And so I think molecular assembly has the ability to be one of those pieces of evidence. And you have to develop them all and start somewhere. So I think, yeah, using this as just the only way to detect life, I would agree. I don't think it's enough. But I think it can be one piece of evidence to combine with maybe atmospheric disequilibrium or other factors to then answer the question of, are we seeing life there?
Okay, Andrew, you had a question.
Okay, yeah. So thinking about, you mentioned all the difficulties of getting the LCMS stuff actually flying to that hardware, that's not quite feasible at this point.
Yeah.
But soon we'll be bringing a lot of samples back to Earth. So, for example, like the OSIRIS-REx mission just brought meteorite samples, and then presumably sometime in the next decade or so, we'll bring samples back from Mars. So, if you were to get those in your lab, would you recommend using LCMS, or do you have any thoughts about that? Like, how we would look for agnostic biosignatures and return samples?
Yeah, I mean, I'm definitely a big fan of LCMS. That's what I use, and I think being able to get those molecular ions is really advantageous, and I think not having to do derivatization is really useful. However, there's a lot of things you can only measure with GCMS as opposed to LCMS. So I think, I mean, they were actually analyzing the OSIRIS-REx samples while I was there, which was really cool to see. And what they're doing is, I mean, there's no reason to just pick one. Like, we don't have to always make that choice because a lot of these sample sizes that you need are really small. So you can run the whole suite of different things. You can measure isotopes, you can do things with LCMS, GCMS, all these different tools, and that gives you the best sample characterization. You can't do everything with one method, so relying on multiple methods, I think, is actually a strength.
Okay, with that, let's thank Josh again. See you next week.